Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
We often picture the tiny, intricate machinery inside our cells as, well, static. Like fixed structures in a factory or rooms in a house, each with a set purpose. But what if one of the most fundamental factories in our cells, the nucleolus?
Yeah.
Yeah, the place responsible for building life's essential machines, the ribosomes.
Right, the protein builders.
What if it isn't just a static blueprint? What if its very geometry is dynamic, adaptive, and actually dictates how fast and efficiently it churns out those crucial building blocks? Welcome to the Deep Dive, where we take your sources and unpack the most fascinating insights. Today, we're diving into some really groundbreaking research published in Nature by Pan and colleagues. It seems to reveal how the nucleolus's structure isn't just, a result of its work, but a core active driver of how well it works.
Exactly. Our mission today is to explore how new super-resolution imaging and some really cutting-edge genetic engineering are showing us the nucleolus is this remarkably organized factory. We're seeing different stages of ribosome assembly happening in very specific compartments. And as we'll get into, even subtle changes to the architecture, the layout, can dramatically impact cell function, even evolution.
Wow.
Yeah, it really pushes our understanding. We're moving from seeing cell organization as passive, where structure just is, to an active view, where structure actively does things.
Okay, so let's unpack this a bit. Before this new research, what was the general picture of the nucleolus? What did scientists basically know?
Well, at its core, the nucleolus is the primary site for making ribosomes, ribosome biogenesis. It's literally where your cells churn out those protein-making factories. And historically, we've pictured it with three distinct layers kind of nested. The fibrillar center, or FSE, then the dense fibrillar component, DFC, and finally the granular component, GC. You can sort of visualize these different specialized departments within a big manufacturing plant, right? Each handling a specific part of the assembly line.
So we knew these departments, FC, DFC, GC, existed, like rooms on a floor plan. But what was the big mystery? Was it about what they did or just how they looked?
That's precisely the question. That's what this study really tackled. For a long time, it just wasn't clear if these layers were just passive reflections of the processes happening inside. Maybe like different colored paint on factory walls, just showing where different activities were located.
Sure.
Or was it more fundamental?
Meaning?
Meaning, did their very existence and how they were arranged actively choreographed the different steps of ribosome assembly? Was the structure itself essential to how the work got done efficiently? That was the big debate.
Okay, I see. So here's where it gets really interesting then. How did Pan and colleagues actually manage to see inside this factory with so much detail? What tools did they use?
This is where the innovation shines. What's really fascinating is their combination of techniques. It wasn't just one thing. First, they used something called live POLEM imaging. That's a type of super-resolution microscopy. So imagine zooming in so far you can see individual proteins like fibrillar and RPA194 at the nanometer scale.
Wow, like seeing individual workers moving around.
Exactly. Not just blurry shapes. And then they combine that with FlamSec pulse chase. It's a metabolic RNA labeling method. This basically let them track the brand new nascent ribosomal RNA bits, the pre-RNAs, as they move through the nucleolus, following the raw materials getting processed step by step.
So they could see the workers and track the products moving through the assembly line. That's powerful. What did this combination actually reveal about where everything happens? Did it confirm the department idea?
It confirmed it, and with amazing clarity. Their analyses, looking at both space and time in human cells, showed this really precise, compartment-specific organization. Think about those pre-RNAs, the raw materials. They found that the precursors for the small ribosomal subunit, the SSU, they kind of linger or held specifically at the boundary, the interface between the FC and DFC.
Okay, so like a specific checkpoint.
Exactly, like a specialized quality control station for the small parts. Meanwhile, the processing for the large ribosomal subunit, the LSU precursors, those mostly moved on deeper into the GC regions. like a whole different assembly line further down the hall.
Right.
So, yeah, it means these layers aren't just labels on a diagram. They're truly functional workstations doing specific sequential jobs.
That sounds incredibly efficient, like a well-oiled machine. But cells aren't always operating at peak performance, right? What happens when things slow down? Maybe if the cell isn't dividing quickly, does the structure change?
That's a really important question they looked into. They examined slow proliferating cells. Cells may be under stress or just not actively growing. And they saw something critical. There was a problem, an impairment, in processing that small subunit, the SSU, specifically related to a part of the pre-RNA called the 5' external transcribed space, or 5' ETS.
Okay.
And this processing slowdown wasn't isolated. It came along with a noticeable structural change, a remodeling of those FCDFC compartments. The layout of those quality control stations actually changed, and the outflow of the finished SSU slowed down too. But here's the kicker, the really compelling bit. They didn't just observe this, they directly messed with the 5' ETS processing. They deliberately introduced a bottleneck at that specific step using molecular tools.
And what happened?
It partially replicated those structural changes in the FCDFC.
Wow, so tweaking the process actually changed the structure.
Exactly. It strongly supports this idea of functional interdependence.
Yeah.
The processing step and the architecture are linked. The factory's layout isn't just a consequence of what it makes, it actively influences production. It's a two-way street. Form and function are deeply tied together.
That's a major insight. The geometry itself is functional. It's not just a container. Did they push this further? Like, look at it from an evolutionary angle. Is this complex structure maybe better than simpler ones?
They did, and this part is really remarkable, a real aha moment, I think. They compared human nucleoli, which had these multiple layers, with those in amniotes. Think zebrafish.
Anomniotes?
Yeah.
Fish and amphibians, right.
Yeah.
No amniotic egg.
Correct. And zebrafish have what are called bipartite nucleoli. Basically, their FC and DFC compartments are merged, not separate distinct layers like ours.
Okay, a simpler design.
A simpler design. And they found these bipartite nucleoli showed a different pattern for that 5' ETS processing step, and crucially, slower overall pre-RNA movement. Their ribosome assembly line was less efficient.
Interesting. So simpler structure, less efficiency. But correlation isn't causation, right? How did they actually prove the structure caused the slowdown? Did they, change the structure?
This is where the experimental design gets really elegant. They use CRISPR gene editing, the molecular scissors in the zebrafish cells, and they use it to introduce an FCDFC interface. They essentially built that separating wall into the normally merged compartments in the zebrafish nucleolus.
They engineered the more complex structure into the simpler cell.
Precisely. And guess what happened? This structural change sped up the processing efficiency. Specifically, it accelerated that 5' ETS cleavage step, the very step that was slow before, and the one linked to structure in human cells too.
Whoa.
Yeah. It directly causally links the geometry to the function. It strongly suggests that when amniotes like us evolved these nested FCDFC compartments, it was a specific adaptation to optimize pre-RNA processing, to make our cellular factories much more efficient.
That makes evolutionary sense. It's amazing how these things get fine-tuned.
It really is. It shows how structure isn't just arbitrary.
So let's pull back to the bigger picture then. What does this all mean for cell biology and maybe even beyond? This feels bigger than just understanding one tiny organelle.
Oh, absolutely. If we connect the dots, this study really elevates our whole view of nuclear geometry. It's not just a passive feature you see in diagrams anymore. It's an active regulator of making ribosomes. Kind of like, how the way thylakoids are stacked inside chloroplasts isn't just for storage. It actively tunes the efficiency of photosynthesis.
Ah, good analogy.
This work shows the physical layout of the nucleolus isn't just where things happen, but it profoundly impacts how they happen and how fast. Structure is function, basically, deeply intertwined.
And the way they figured this out was pretty cutting edge, too, wasn't it? You mentioned the combination of techniques.
Definitely. It was the synergy, really. Having the live palm imaging for those super detailed maps, the SLAM-seq to track the RNA kinetics over time, and then the CRISPR engineering to directly manipulate the structure and prove the cause and effect link, that combination is a massive leap forward.
Right. Not just one trick, but the whole toolkit.
Exactly. And it's worth mentioning, this whole project was a big collaborative effort within a Shanghai consortium. They pooled expertise in microscopy, RNA mass spectrometry, zebrafish genetics, and even computational modeling to translate all that spatial data into kinetic models.
That really highlights how modern science works, doesn't it? Bringing different fields together.
It does. You had different lead scientists focusing on imaging, CRISPR engineering, the evolutionary parts,
Uh-huh.
all guided by Ling Chen integrating the concepts. True team effort.
So, looking ahead then, what doors does this deep dive open? Are there potential applications, maybe? You mentioned cancer earlier.
Well, it sets a really powerful precedent. The idea is, if we can understand how architecture controls function in the nucleolus, maybe we can apply that to other phase-separated organelles in the cell.
Those little membraneless droplets that organize things.
Exactly. They're involved in all sorts of processes. If we could subtly tweak their internal architecture, maybe we could rewire fundamental cellular pathways. And yes, regarding cancer, we know the nucleolus often changes size and activity in cancer cells. This is called nucleolar plasticity, and ribosome production is often ramped up. So understanding how structure controls this might, down the line, inform new anti-cancer strategies.
Targeting the factory's layout, not just its output.
Potentially.
Yeah.
Now, translating these kinds of structural edits directly into mammals, into humans, that's still a big hurdle. It's much harder than in cell culture or zebrafish.
Sure.
But the principle that we can actively manipulate an organelle's architecture to control cell function, that opens up a really fascinating new frontier in biology. It's a whole new way to think about intervening.
So wrapping up, what really stands out to you from this deep dive? For me, it's just, wow, how much we're still uncovering about the absolute fundamentals inside our own cells and how incredibly sophisticated and adaptable they are right down to the physical layout.
Yeah, I agree. It really drives home the point that what looks like just structure in a static textbook image is often anything but. It can be this dynamic, actively managed system, a living factory, like you said, constantly optimizing itself, not just a blueprint.
So maybe next time you think about a cell, you can consider this. If the precise geometry of this tiny membraneless factory can optimize something as vital as making ribosomes, what other cellular structures are we maybe underestimating? What else might look like just backdrop, but could actually be pulling the strings for some of life's most essential processes? Something to think about until our next deep dive.